So Dr. Temenoff is on, and so I wanted to welcome her. And all right, yay, she's at the beach. Thank you for joining us, Dr. Temenoff. We're so excited to have you here.
Thank you. I just want to make sure you can hear me.
Yep, we can hear you. All is good. So Dr. Temenoff has a really incredible background, and I'm going to just go over a little bit since we only have a half hour. I could read about all of her accomplishments for quite a while, but just a little foundational piece. She completed her PhD and postdoctoral fellowship at Rice University in tissue engineering and orthopedic biomaterials. In 2005, she joined the faculty at the Coulter Department of Biomedical Engineering at Georgia Tech and Emory University, where she's now a Carol Ann and David D. Flanagan professor. Dr. Temenoff is also currently the deputy director of an NSF Engineering Research Center in Cell Manufacturing Technologies, and that's what she's really going to focus her talk on today. And you will probably hear the acronym CMaT, and that does stand for Cell Manufacturing Technologies. She was named the Associate Chair for Translational Research in the Coulter Department in 2019. And scientifically, Dr. Temenoff is interested in tailoring the molecular interactions between, practice this word a lot, glycosaminoglycans and proteins or cells for use in regenerative medicine applications. Her lab focuses primarily on promoting repair after injuries to the tissues of the shoulder, including cartilage, tendons, and muscles. So Dr. Temenoff has received many awards, but the one that I'd like to point out is due to her dedication to education, particularly in undergraduate biomaterials education. And she's a textbook author, so she understands that, understands pedagogy and kind of the learning layers that are involved. So I'm going to pop into the chat some of her other awards and you guys can take a look. But without any further delay, Dr. Temenoff, thanks so much.
Thank you. Well, it's great to be here this afternoon, and yeah, I'm trying to cheer it up a little bit. It's rainy here, so hopefully you can at least enjoy the background. I'm going to go ahead and share some slides, and we'll just talk a little bit about CMaT and kind of our what we would hope to pair with you to do. I have so many options to share now. All right. All right, can you see the slides and my red laser pointer? All right, excellent. Okay, so thank you so much, Kristen, for that great introduction to me and also to CMaT, so I don't have to spend as much time on it. But basically, this is a multi-institutional organization that is headed at Georgia Tech. We also have partners at UGA and Emory, other places throughout the state, and we're really looking to advance manufacturing of therapeutic cells. And this is funded through NSF, and it is what we hope to be a 10-year-long endeavor. And it is a pretty substantial investment by NSF, and it's not only in the area of actually doing research, but we have a very large mandate in the area of education, and that's what I wanted to talk more with you about today. The overall vision is to actually enable the robust and low-cost biomanufacturing of high-quality therapeutic cells. And we believe that this is really necessary to enable access to these really curative emerging technologies to everyone. And just to give you an idea, we are a broad partnership, as I mentioned, centered in Georgia, but we have other partners throughout the US, and actually the dots on the map show our international partners as well. So I like to start out with just talking as we do. So we're about, on the order of 40 to 50 investigators and then all of our trainees. So we're a group of at least 200 people, and we try to bring people together a couple times a year. And one of the things that we like to really talk to the trainees about is why are we here as a center? And really, we're here to help patients. And so to talk about this, we believe that by working together, we can really make these really new and emerging therapies, again, accessible to everyone all over the world. So we have a number of speakers at our retreats every year, and these are just a few examples. And these are actual patients that have been helped by cell-based therapies. And so we take cell-based therapies and define this broadly, but largely the ones that have been FDA approved are immunotherapies that you may have heard of, in which you take your own T cells and engineer them and inject them back into the patient so that they can fight cancer. So this is Emma over here, and hopefully we can play a video and you can hear a little bit more about her story later on. But she was, very, very sick with childhood leukemia, and now over 6 years that she's been completely cured based on these T-cell based therapies. Austin is another example, and he came and talked with us a couple years ago. Again, these children wouldn't be here today if it weren't for these therapies, and they're our continual inspiration. Our advisory board members include Amy Kappen, the mom here, and this is her daughter, Sophia, that unfortunately also had childhood leukemia and didn't make it. She passed away waiting for the cells. And so the speed at which we can really make these therapies available is very important as well. And this is Doug. He is actually almost patient zero. He is really the first patient who is in the clinical trial for these now FDA approved therapies. So we're very, very lucky to have these people as a part of our larger family, and we really continue to take inspiration from them. So to provide a little bit of background, about 4 years ago when we started CMaT, there was a lot of exciting news coming from industry in the area of these cell-based therapies. And I mentioned that there was, particularly in the area of immunotherapies for cancer, there was 2 big FDA approvals, and those are kind of shown up here, but there's also many other types of cells that are being investigated for and going through clinical trials now. And so just to kind of give you the perspective in the past 3 or 4 years since then, we now have thousands of ongoing clinical trials. These are just a list of therapies that are very likely to be approved either in Europe or in the US coming up. And we've actually had now 4 therapeutics, cell-based therapeutics for various kinds of leukemias. So this is just a highly expanding field. And what we really want to do with CMaT is not only, propel the field scientifically, but we also want to make sure that we have a workforce that's able to manufacture these cells. And actually, very specifically, we want to do this in Georgia, and we want to help patients in Georgia. And so that's why we're really interested in partnering with groups like you. This is just another graphic that just kind of shows the potential economic impact of these types of therapies, and that we really need to be able to scale up and manufacture these at a large scale. So the major issue right now is the cost of these therapies. So these are curative therapies. So these are not things that necessarily have to be injected more than once or maybe twice. But preparing these and making sure they're safe and getting them back into the patients is costly. And so what we're really trying to do is kind of expand the manufacture of these cells, because if we get better at manufacturing them, we're going to drive down the cost. So what is the impact on the workforce that we foresee? Clearly, there's a lot of interest in industry in building very large cell manufacturing facilities in various locations, including in the state of Georgia. So we really think that this could drive a number of high-paying jobs in the area of biotechnology. And this is not just for PhDs. This is for people working on the floor to actually manufacture these cells. So we're really looking at all levels of job creation. And just to highlight this, this advanced biomanufacturing is something that's been on kind of the national radar for some time, and we're trying to focus it particularly in outreach at K through 12 as well as community colleges on cell-based therapies, and that's what CMaT's educational mission is. So what are the challenges and barriers that we see in scaling up and manufacturing these potentially life-saving cell therapies? So the main problem is to get enough of them and to get them as a reproducible, high quality product. And the real barrier to this is that the product, you know, if we think of the complexity that we've just seen with the vaccine rollout, cells are even more complex. They're living entities and they change every time we do some sort of intervention with them in the manufacturing process. So this is a very dynamic process, and we need to have people that really understand the complexities of cell culture to really be able to make and culture large numbers of these cells. So, you know, they're very complex and powerful. You can think of them as depots of multiple drugs at once. But the problems are that every source is different, and then every user is different. And also, you know, this part down here, that we have a rapidly perishable product. And so it's not only about manufacturing them, but it's also about getting them out, much like the vaccine distribution, to areas that, you know, it may be difficult to maintain cold chain and so forth. So we're really looking at, from the technology perspective, kind of addressing both biomanufacturing as well as supply chain and distribution. So one of our collaborators, who is part of the first clinical trial, says that, you know, currently for cell manufacturing, this is like 1906 for the automobile industry. So we're really just starting from the beginning. We don't even know really quite how to build the factories, because we're not sure how that's affecting the quality of our product. So it's really a partnership between scientists and industry, which is what CMaT is trying to do, and then bringing in workforce to be trained to work in these complex environments. So what do we see as the grand challenges in cell manufacturing? The lack of reproducibility and standards are really kind of a difficulty right now. And this goes to the second point, which is, you know, we don't fully know for all of our therapies. For some of them we do, but not for all of them, what quality attributes makes a cell safe and effective. So we don't even fully know what to measure as we're moving through our process. How to separate good and bad cells. And then how to do this in line during manufacturing. So you can think of a big reactor, and what would we measure, and what could we control? So all of this biotechnology, these sensors, and this system that's been set up for producing other types of biological molecules, we need to really rethink in the context of cells. We're currently at very high doses for many of these cells, so we really need to scale up to very large billions of cells from a single small donor sample. And then, you know, how do we predict which batches are good and bad early on? So these are very costly processes, and if we know a batch isn't going well, most manufacturers would prefer to get rid of it and start again. And we need to do that very early on in the process. So, and then assuming we could even get all of this information and have good batches, then we need to go back to that idea of purifying, storing, transporting, and making sure we don't lose quality in that process. So again, a very dynamic process, and one in which we don't always know right now. And so, you know, going down here then, though, every time we talk to industry, and I'll talk a little bit more about this, they really mention that another critical barrier is a lack of a trained cell manufacturing workforce that really understands these sorts of complexities. So what do we think we could do if we could get to this idea of quality-driven manufacturing? So this is a high-risk product in that it's very expensive for these very large batches. And so we think we could reduce risk to the company, financial risk, but also risk to the patient if we had a better idea of what made a good cell. And then we also think that we could potentially reduce the dose, because if we had higher quality cells, they might be more effective at, for example, killing the cancer, so we wouldn't need as many of them. All of this would play together to reduce cost. We also think that we would be able to inform regulatory processes. So right now, as we've just experienced, the FDA approval process can be long and arduous. And we think that we could inform them about what are the most important things to show safety and efficacy of our therapies, and that they could get approved faster. And then, all of this kind of is the idea of we need to develop standards and best practices in the field. And we need data to do this. So this is one of the things that we're trying to do within CMaT is understand if we change certain parameters, how does that affect the phenotype of the cell that's being produced? So how did CMaT start? We started, this is probably about 6 years ago now, in bringing a large group of people together. Obviously, this was pre-COVID so that we could all actually be in the same room and chatting. And this is industry, academicians, clinicians that are using these cells and government all came together to build a roadmap for what would be needed to move forward the field. And this was interestingly, even before the first FDA product came on the market, was our first roadmap. And so we developed this roadmap that has to do, again, with these various technologies, but also, what we call the industry foundation, which is standardization, workforce, and looking at these supply chain elements as well. So we came out with this ultimately in 2016. It's been updated a couple times since then. And so we have currently now a roadmap to 2030. And just to give you a peek inside, this is some specific things that we think need to be done in terms of cell culture, advances in processing equipment. And you don't see it here, but these are target dates. And so this is the kind of thing that we've been talking about, again, driven by industry. But probably the most important part is that we have a whole subsection on workforce development. And what I'd like to just highlight here is that, again, this is not just for graduate programs, but that we have goals for really all levels of education. And again, that industry is very, very interested in having a workforce that's educated and wise to cell culture at all levels. One other aspect that I wanted to point out is that, kind of in parallel with this multi-institutional endeavor that is CMaT, we have a specific center at Georgia Tech called the Marcus Center. And what it really is actually a pilot manufacturing facility. So this is a GMP level suite here, and this is for process development and validation for some of our new technologies. But basically what it also provides is a small training suite. And so what we hope to do once we're allowed, again, is to have, if you're interested, we're trying to organize some field trips and things that you can come in and see this facility and see the types of environments that people in the field are actually working in to produce these cells. It is quite constraining and it feels very different than even if you've done, traditional cell culture in a cabinet. So the overall goal for CMaT is to bring what we call all of our stakeholders. And this is a whole bunch of different engineers and scientists, but also we believe just equally important is, regulatory and standards, industry, reimbursement and patient advocates, but also our workforce experts. And we really believe that all of these people are needing to come together, and we're trying to provide this place within CMaT to really get to this high quality cells at a lower cost. So to break it down for those of you that are interested a bit more in the science, what we are doing here is taking input from all of these external stakeholders, and we kind of distilled it down into 3 scientific questions, and they're actually shown in the black here. So it's what to measure to ensure quality. So we're still trying to figure out what a high quality cell is. And then how to measure it, particularly in line, and then actually how to scale. So what kinds of bioreactors and other technologies would we need to get to those really large number of cells? And so we have researchers that are centered in each of these questions. And we've actually had them apply their technologies into several different therapeutic cells. So I mentioned the most about these T cells. And so we spend a lot of time on that because they are the FDA approved product. But we also have other cell types that we see are emerging and that might have FDA approved products relatively soon. So we're also working on some of those as well. And so the outputs are obviously, we want to have new tools that would push industry based on that roadmap. Sensors, better bioreactors, that sort of thing. But we also want to use our data to get to that what is a good cell, and push the national standards boards to come up with standardized practices that would, again, go across all of our industry members and really promote high quality cells. And then finally, using all of this information to do training at multiple levels. Another way to look at this is, that underneath CMaT, we have currently, more than 20 industry partners. And these are, people that actually produce the cells, all the way down to people that produce equipment like bioreactors that would interface with the cells. And we're trying to bring these all together as well. And we believe that these are equally important to our scientists to get to all of our goals. And then just one slide on some of our standardization efforts, just to show you kind of a peek at where we are in the industry. These are some things that our scientists have been involved with at the federal level, trying to get standards. And so if you look at this, I mean, these are, very basic things. Like, so how do we characterize these cells? What are cell manufacturing equipment? So this is just, again, to show you that we're very, very early in this industry, and we're really trying to build the industry as we're trying to create a product, which is a really interesting place to be. So I just wanted to spend a couple minutes on the workforce side. So, as I think you appreciate, we have multiple different, what we call pillars. So we have scientific pillars, we have workforce pillars. So the goals for our workforce pillar is really to, again, work at multiple levels to create new curriculum, but also to pair with industry to do training programs and other things to really get them to the entry-level workforce that they need. And this is a fancier diagram, kind of showing the same thing, but that we hope to work with you and other partners to look here particularly for building an entry-level workforce, and to stimulate interest at the K through 12 levels for people that maybe didn't even know that this could be a career, because we really do believe that, given this large number of new products on the market, that this is really going to be a place that they might be able to be employed in, particularly in the next 10 years. This is an up and coming area of biotechnology. So I'm going to move on to, in closing, hopefully play a video for you. So I'm going to go next, and I'd like just somebody to make sure that they can tell me, like, give me a thumbs up if you can hear this. Can you hear anything?
Not yet, or no. But there should be something at the bottom of your screen, maybe at the top with 3 dots.
Yes. Aha.
Patients that we're treating on this clinical trial have absolutely no other options left for them. These are patients who are unfortunately destined to die of their disease, and in a fairly short amount of time.
Can you hear that?
Whitehead 554.
So Emma is incredibly matter of fact about all of this stuff. This was a child who had had her leukemia come back twice. The parents were looking for a miracle.
What we've learned how to do is train the immune system to recognize and then kill tumor cells.
It's a procedure where we collect their T cells, and they are infected with a virus that will genetically change them so that they will now see and react against their leukemia cells.
We call those modified immune cells serial killer cells. Each infused cell can kill more than 1,000 different tumor cells.
Emma was given her T-cell treatment, and within a few days, she was very sick. She had breathing difficulties, she had blood pressure difficulties. We knew that she could not have gotten any sicker without actually dying.
But then a remarkable thing happened. The T cells were growing, they were starting to fight the cancer.
Within hours, Emma's fever disappeared.
It was like the calm after the storm. The clouds went away. and she woke up and there was no leukemia. When that child survived, it was, of course, an amazing event.
So I like to close with that because that was the little girl that you saw at the beginning who's much older now and is doing super, super well. So this is really, this is why we do this. You know, we want to do this for millions of Emmas. So we would love to have you or your students be a part of this. And so I just wanted to close with this, which I believe will be circulated to you. If you're interested in being a part of CMaT, particularly this summer, we do have a summer experience for teachers, and it will be virtual this year. It will be about a month. There's actually some time for you. You will be paired with a researcher, but there's also time for you to develop some curricular activities to introduce cell manufacturing to your students. So, you know, again, this is really a hugely important aspect of CMaT. We would love to have some of you participate, you can use this contact information, and I think Kristen will send this out as well. Mm-hmm. So I thank you for your time, and I'm happy to answer questions.
I've got a few hundred. Looks like you're going mostly after differentiated cells. You don't work with undifferentiated cells, I suppose. That's so pretty controversial.
Well, one of our cell types is actually an adult stem cell, so it is not completely differentiated, but yeah.
Getting any resistance from that?
No, these are, you know, again, they're derived from bone marrow, okay. And so it's not, yeah, we've stayed away from that.
that is the, if you can find stem cells, the world is open. Geez. So basically, it's a whole lot more than just simply growing tissue to cover burns. No question about that.
Amazing. So it was interesting. You did draw a parallel with supply chain, and that these cells are even more delicate, so to speak, than the ingredients in the vaccines. And so it's kind of been, we've never spoken, I think, at least in my lifetime, so casually with vaccine terminology and some of the public health terminology we use today. So it's kind of nice for kids because it's in a language that they're immersed in now, and so something that they're used to connect with. So understanding that the vaccines have a very short shelf life, they need to be used. This is a great time to draw some comparisons to the difficulties with this type of treatment, with the personalized cell treatment. Are there storage needs similar to the COVID storage needs?
Yeah, I always, my current nerdy joke is that, you know, I cared about cold chain supply storage before it was cool. Yes, actually, very similar to the Pfizer vaccine, -80. You know, we would have to transport on dry ice, those sorts of things. So it's really, we actually have one project trying to think about ambient ambient temperature transport of cells, which would obviously really open it up worldwide. But it is absolutely a concern. Yeah.
I feel like the cold chain piece would be really interesting to a whole other group of students too, that, yeah, interested in science, but no interest in being in a lab but they get the basic concepts and so on. It'd be really neat to bring somebody in to talk to classes or about how to pursue that or pursue some experiences in the summer for cold chain. Do you have any connections regarding that or suggestions for teachers maybe? This is totally not a planned question, so throwing you on the spot.
Yeah, I mean, yeah, actually we do. We have some supply chain modelers. Actually, if you're talking about, you know, completely non-wet lab, that are really looking at, you know, the constraints of how far you can travel in a day and that sort of thing. And so we could set you up with that perspective. We also have some people at Georgia Tech that have worked on mainly for proteins and non-cells, have worked on this for quite some time. So I'm happy to connect you.
And with UPS headquartered here too, we're a huge supply chain state anyway, but they have a whole healthcare division, UPS.
Yes, yes, and we're talking with them as potential partners within CMaT as well. So yeah, it's very exciting. I think you know, we try to pull a silver lining from the past year, and that may be one of them.
Anyone else have questions? I always feel like I dominate. I will give just kind of a heads up that we are looking and getting closer, I'm going to knock on wood, to a biotechnology technician assistant certification exam that your students would be prepared to take after finishing their pathway, that will actually be recognized by industry. So we're working very hard on that to have everything pass and get the funding to then make that accessible to you guys. So just kind of stay tuned for that. But that kind of experience would be ideal to lead into workforce for cell manufacturing technologies.
Yeah. No, I mean, I think the thing that I've been struck by over and over again is just that there's a place for people in this industry, you know, regardless of what you want your exit strategy from education to be. So I think that's really, really fantastic.
I've noticed in the seminars that I've attended with CMaT too, that there's really a strong need for the technician, just a certified technician, which you can get through a 4-year degree plus a certification, or you can get it through a 2-year technical college, which we have several of around here. And then I think it's in Madison, Wisconsin. They have a really strong biotechnician certification exam, and everything we're covering here, which is, you know, a basic level, is foundational to that. And then if your school has that biotechnology pathway too, you're halfway there. Your kids are halfway there, if not more. So we're on a good track, and Georgia has a really, really heavy footprint in cell manufacturing.
Yeah, and you know, really, when companies come in to talk about location, they want to know what our workforce training plans are. And so, you know, we want to be the cell manufacturing hub of the world, and we need your help. I think there's just a couple questions in the chat. I'll try to be brief. The HeLa cells we do not use. They are a derived cell line. We use primary cells from donor tissues, which makes them much more difficult because every donor is different. But it's much more like what you would see in clinical applications. And then neurofibromatosis, I'm actually not familiar with, but there's been a lot of, particularly these adult stem cells, use for a variety of anti-inflammatory applications. So it would surprise me if someone hasn't tried it for that.
Well, thank you so much, Jana, for your time and your insight into cell manufacturing technologies. And I'll definitely pass along the opportunity for the summer experience as well with your contact info. And the nice thing about this, again, with it's one of the blessings coming out of COVID is being that this is virtual, it opens it up. So if you
Exactly.
wouldn't have been able to travel to Georgia Tech or UGA, you'll get to at least get the virtual experience in that lab.
Yeah, so definitely, again, we're going to do a virtual pairing, so you'll get a semi hands-on experience, a virtual experience. Hopefully this gives you an opportunity to see what we're doing. So thank you again, and enjoy the rest of your evening.